Okay, sorry for being late. I had trouble logging in. Well, first confirm that you guys can hear me, please.
I can hear you.
We can hear
Great.
you.
Are there any questions? All right, I'm not hearing anything, so I'm assuming there's no questions. I saw a number of emails, but I haven't looked at them today. I did answer emails yesterday, though. What are we on? All right, today is January 26th, and we're going to be covering chapter 4. Let me share my screen. I think we started chapter 4. Oops, wrong one. Let me open that and come back to the syllabus. In the lab today, I'm going to explain about the unknown project. And some students get confused about it. So please pay attention to my discussion about the unknown project. You are going to be doing an unknown project, but it's going to be all online. And we're going to pretend that I give you a bacteria, and then you're going to identify it. Any questions about any of that?
When will you be giving us our numbers for it? Like tonight or tomorrow?
If I am, oh, wait a minute, wait a minute. I might be confused here. Actually, I'm only supposed to tell you about reading the unknown project instructions. This is not going to start until January 31st, where I will be discussing it actually next Tuesday. So sorry, I didn't check that very carefully. I just saw unknown project. We're not really going to discuss it until next week, next Tuesday. So I'll be giving you, hopefully, your number late Tuesday night. It might be after midnight, but hopefully it'll be around 10 p.m. because it'll be after the lab. So it'll be, the earliest I could get it done would be 9 o'clock, but it might be, it'll probably be closer to 10 o'clock and maybe as late as midnight. Okay?
Thank you.
All right. All right. If there's no further questions, let's go to chapter 4. I should warn you that I heard that the IT department is going to be installing an update. I think it's for their security, and it may interfere with the network, in which case I might lose connection. It's not going to be tonight, but sometime in the future, they're going to be doing this, and I will try and inform you when it happens. I don't remember the exact date. It's sometime in February. And I couldn't believe that they're doing this. It is at night, so it will affect us. And they said, and you will have interruptions for a few minutes during this time period. And it will interfere with Clark College, which I initially go to Clark to log into Zoom to then start Zoom. So it may not interfere with Zoom. So if I get started and we're just doing Zoom, it shouldn't be a problem. However, if I want to get to Canvas during that time, there will be interruptions, at least for a few minutes at that time. I don't usually get into Canvas. Let me, while I'm here, I don't need that page. Hopefully I didn't close the browser. Yep, I did, darn it. I was going to try to show you. Gosh, that thing is just in the way and I can't move it. There it goes. I was going to try and show you where the instructions are for the unknown project that you need to read in Canvas. I think it's under week 5 lab. See, when I was in that Zoom page, I got there from the course page and I didn't realize that it was in Canvas, but it was. Let's scroll down. I think it's week 5 lab. Yeah. So the unknown instructions, read that, and then we'll discuss that next Tuesday. You could read all of these other things, but I'll talk about them on Tuesday. And you do not need to read any of this material other than the unknown instructions. You would need to read the other material when you start working on your unknown, but you're not going to do that for a while. All right, let me go to our lesson. And so we had talked about damaging prokaryotic cell walls, and they have different names for the cells. If you do it, you don't need to know the names. But you should know that gram-negative cells are not as susceptible as gram-positives to certain antibiotics like penicillin G and certain detergents. I forgot the name of the family that's similar to penicillin. But penicillin G and penicillin V do not work on gram-negatives because the outer membrane prevents the antibiotic from getting inside the cell and damaging the cell. And some detergents too, but that doesn't mean that gram-negatives are not susceptible. We do have antibiotics that only work on gram-negatives. Polymyxin B, now I remembered it. I remember last time I was discussing it, I couldn't remember. Polymyxin B is an antibiotic that only works on gram-negatives. And then there's other antibiotics that can penetrate the outer membrane of the cell wall, including streptomycin. Streptomycin is a lipid-soluble antibiotic, and so it can get right through the outer membrane of the cell wall. So let's talk about the plasma membrane in eukaryotic cells and in bacteria. The plasma membrane is a phospholipid bilayer where, let me blow this up. Where the cell membrane is composed of one phospholipid layer and then a second phospholipid layer where the heads of the phospholipid are facing outside the cell, and on the other layer, the heads are facing inside the cell, so that the heads face the water in the cytoplasm of the cell, and then the outside of the cell is mostly water. If you don't know, most living organisms are about 70% water. That's true of humans too. And you can just think of this as a red blood cell, and the blood is mostly water. And so the heads of the phospholipid are facing the mostly water in the blood. And then the fatty acid tails of the phospholipid are together away from the water. So that's the main molecule in the plasma membrane, also called the cell membrane. There are other molecules in the plasma membrane, like certain carbohydrates, like glycoproteins or peripheral proteins. and other proteins. There are 3 different types of proteins in the plasma membrane. There are peripheral proteins, integral proteins, and transmembrane proteins. The peripheral proteins are proteins which are mostly on one side of the membrane or another side. Okay? And that's why it's peripheral. It's peripheral to the membrane. The integral proteins and the transmembrane proteins span the membrane. Probably the most important one is the transport protein, which actually pumps a molecule from one side of the membrane to another side. Any question about any of that? Okay, so the cell membrane is a lipid bilayer composed of phospholipids. There are other molecules in here. I don't have it shown. In animal cells, we have cholesterol in the cell membrane. And of course, there are carbohydrates and proteins. Come on, let's move. Now, the plasma membrane is actually a fluid, and we have the fluid mosaic model to describe the plasma membrane. The fluid mosaic model states that the cell membrane is a fluid of phospholipids and proteins. And the membrane is actually, at least at our body temperature, is actually as fluid as olive oil. And it's always amazing to me to think when I hold up my hand, what's holding my cells together is the plasma membrane, which is a fluid. And it's just always amazing to me when I think about that and I look at my hand that it's being held together by a fluid. Proteins can largely move about in the membrane to function. Not all proteins move, but many of them do. And those of you who have had A&P, you know that when a receptor binds, not a receptor, a ligand binds to a receptor, the receptor protein, they tend to move together in the plasma membrane before they're brought into the cell to give the signal to the cell. So proteins can move about to function, and that's both spinning around and then just moving laterally. The phospholipids can also move laterally and then spin around in place. And there's nothing stopping the phospholipids from moving around. It's just there's so many of them. I don't think anyone's ever bothered to watch about where they go, but they can move because the plasma membrane is a liquid. Not all of the proteins move. Some of the proteins are anchored in place. There you go, previous, I don't have a picture of that. And that would be a protein which is anchored to the intracellular matrix. That's a matrix of proteins making the backbone of the cell. And if that is connected to a protein in the membrane, then that protein is not free to move about. It's fixed in place. Another way that a protein could be fixed in place if it is anchored to the extracellular matrix, like the extracellular matrix in our bone is largely, what is the material of the bone? Calcium and something. I'm sorry, I'm forgetting. Whatever the structural protein is in bone. But that extracellular matrix can bind to a protein in the membrane, and then that protein is not free to move about. Okay, I lost my mouse here. Where is my mouse? There it is. So from someplace outside the cell binding to this protein. But many proteins, most of them in the membrane, are free to move about. The plasma membrane shows selective permeability. That means some molecules can move through the plasma membrane, other molecules cannot. When we're talking about large molecules, no large molecule can move through the plasma membrane one way or the other without the help of transport proteins. All large molecules cannot move through the plasma membrane unless there's a transport protein to move it across the membrane. Ions move either very slowly across the membrane or they do not move at all. Lipid molecules can move through the plasma membrane because this is a phospholipid. It's made of lipids, and lipids can get right through it, and a lipid outside the cell can move into the cell and vice versa. Smaller molecules can also cross the plasma membrane. The two most important are oxygen and carbon dioxide. I think you all know that our cells require oxygen, and it will diffuse through the blood, across the plasma membrane, and into the cell where it's needed. And then the cell produces CO2, and it can diffuse across the plasma membrane out of the cell and into the blood. And then the blood will carry that carbon dioxide to the lungs, and then we'll breathe it out. Water is also another molecule that is small enough that it can cross the plasma membrane. I'm trying to get a big picture here of the plasma membrane, and it looks like I'm just going to have to blow this up. What happens is water waits for a phospholipid to move over, and then there's a space for the water to come in, and the water will come in and just wait right here, meaning it's got this space open so the phospholipids can't move in, and it can't move through the second bilayer, but what it's doing is it's just waiting for this or that one to move over, and then the water can come through the plasma membrane. So water can move, I don't know, sort of slowly through the plasma membrane, and if there is more water outside the cell, it'll move into the cell, and if there's more water inside the cell and less outside the cell, water will move outside the cell. And like I said, it can move through the plasma membrane, but not very quickly. If for roots of plants, for example, they have transport proteins to help move the water, or maybe channel proteins to help move the water outside the root into the root cell. Okay, and that's to move water quickly from one side to another. Obviously, if you have less water outside the plant will lose water, and the plant will start shriveling. But small molecules can move across the plasma membrane. Well, what about other molecules that we need in our cell, like glucose and fructose and amino acids? They are large enough that they do not move across the membrane without the help of a transport protein. However, the cell wants those molecules, and so it will make a transport protein to move those molecules across the plasma membrane. Any question about any of that? All right, some miscellaneous things I need to talk about the plasma membrane. For prokaryotes, and nope, it's not for eukaryotes. For prokaryotes, we have enzymes in the plasma membrane for making ATP production. And I'll talk a little bit more about that in a later lesson. For the photosynthetic bacteria, we have the photosynthetic pigments also in the plasma membrane. However, they're in special regions of the membrane, which are folded in among themselves, meaning here's the plasma membrane going like this, And let's go ahead and go with this one. Plasma membrane comes out here and then goes around and then around. And actually there's another one there. And that fold is where we have the pigments for photosynthesis to occur. And they actually have a name. You don't need to know that. Thylakoid or chromatophore. Let me shrink that down now. Yeah, that's actually better. It's an error there. When you damage the plasma membrane, you'll have leakage of the cytoplasm outside the cell, and that will kill the cell. And that damage to the plasma membrane can happen with alcohol, detergents like quaternary ammonium compounds. And I mentioned polymyxin B just a little while ago. That antibiotic does damage the plasma membrane and puts a hole in it, which will cause the cell to die. All right, any question about any of that? All right, movement of materials across the membrane can happen. The movement of materials is critical across the plasma membrane because the plasma membrane is the barrier to inside and outside the cell, at least with the movement of material. Anything that the cell wants, it'll want to move it into the cell. Anything the cell doesn't want, it'll want to move outside the cell. So the movement of material across the plasma membrane is critical. Movement can happen in two different processes. One is a passive process where the material moves down a concentration gradient. That means, like for CO2, it's more highly concentrated inside the cell. Come on, mouse. And the carbon dioxide will move down the concentration gradient, moving outside the cell. The other process is an active movement where the molecule moves against its concentration gradient, in which case you need a transport protein which uses up ATP to move the molecule across the membrane. For example, if you were to eat a high sugar diet containing the sugar glucose or sucrose, which is broken down into glucose and another sugar we're not going to talk about at the moment, because I only want to talk about glucose, the cell will have a transport protein on the plasma membrane, and it will pump glucose outside the cell at higher concentration to inside the cell. And because the concentration is higher outside the cell, glucose can move into the cell without using ATP. Okay, so that would be actually a passive process. I guess I shouldn't have used glucose as my example. And then I gave you the example that you're having a diet high in sugar, but the sugar will be totally digested, meaning 100%. And so when the sugar glucose concentration outside the cell equals or becomes less than the glucose concentration inside the cell will still want that glucose molecule. And then it will pump the glucose molecule across the membrane with the transport protein using ATP because glucose is moving against its concentration gradient. So it's moving from areas of low concentration outside the cell to areas of high concentration inside the cell. And that is an active process. Any question about that? And sorry, I shouldn't have used glucose as my example. Glucose is a weird transport protein, and that is when glucose is higher outside the cell than inside the cell, this transport protein changes to a channel protein, and then the glucose can move directly into the cell without using energy, and that's facilitated diffusion. We'll talk about that in a minute. But when the glucose, says my internet connection is unstable again. I don't know what my problem is. It must be my internet service provider. I'll have to contact them. If you don't hear me, just ask me to repeat. Okay. But when the glucose becomes less concentration or equal in concentration to the glucose inside the cell, the protein will change from a channel protein to an active transport protein, and then ATP is used up to pump the glucose outside the cell, inside the cell. Any question about active processes? All right. When we're talking about passive processes, there are three passive processes. There's simple diffusion, where a solute moves from areas of high concentration to areas of low concentration. There's facilitated diffusion, where a solute moves across a membrane from areas of high concentration to areas of low concentration. And we'll talk more about that in just a minute. And then there's osmosis, where water moves across a membrane from areas of high concentration to areas of low concentration. Water is a solvent, and it's moving the same as simple diffusion, but it is using water is moving across the membrane, so it's sort of a combination of these two. The main thing, these two, but the main thing is osmosis is the movement of a solvent, specifically water, across the living membrane or a non-living membrane. Any question about any of that? All right, simple diffusion is just the movement of a solute from areas of high concentration to areas of low concentration. For example, if we put a drop of dye in the beaker, this is the area of high concentration, and the molecules of dye will be dispersed over time in the water of the beaker, moving from areas of high concentration to areas of low concentration. And as I stated, this is the way that oxygen and carbon dioxide move in our cells. Facilitated diffusion is similar. A molecule moves from areas of high concentration to areas of low concentration. However, in facilitated diffusion, this molecule is moving across a membrane, and to move across the membrane, it needs a channel protein. This picture is not calling it a channel protein, it's calling it a transporter protein. Because the molecule is moving from areas of high concentration to areas of low concentration, a molecule of ATP is not required. Any question about any of that? Glucose is not the best example. It does work, glucose does work that way when the concentration of glucose is higher outside the cell than inside the cell, in which case glucose moves by facilitated diffusion. However, your cell will want every glucose molecule it can get. So it will change from a facilitated diffusion to active transport, and then pump that glucose into the cell until there's no glucose outside the cell. So all the glucose will be digested. But a better example of facilitated diffusion is actually the channel protein for bringing fructose into the cell. If you eat a high fruit diet, there will be a lot of fructose in your gut. Your cell will want it. It has a channel protein or a transporter protein, and that will move the glucose into the cell by facilitated diffusion. Fructose does not move into the cell with the help of ATP, so no ATP is expended to move fructose into the cell. And fructose will move into the cell until the concentration of fructose outside the cell is equal to the concentration of fructose inside the cell. So that means that not all fructose will be digested. Another reason why it's better to eat fruit and fructose than it is to eat other sugars like glucose or sucrose, because all of the glucose will be digested, but fructose, not all of the fructose will be digested. Now, if you're wondering about the digestion of fructose, it's a little more complicated because this cell can take this glucose and then put it into the blood. The blood can carry the, not glucose, take the fructose and put it into the blood, and the blood can carry the fructose to the liver. The liver will pull the fructose out of circulation, out of the blood, and then can start slowly converting fructose into glucose. And then it'll dump that glucose into the blood. And that's why diabetics can eat fructose, because it is digested slowly, not all at once. But the point is that the fructose in this cell will decrease, and then more of the fructose can move in. And you don't really need to know any of that. I'm just telling you for your own information. All right, so both simple and facilitated diffusion transports or channels the movement of a solute molecule, and the molecule moves down its concentration gradient from areas of high concentration to areas of low concentration. Osmosis is similar, except it's the net movement of water, a solvent, not a solute. And the water is moving across the membrane in osmosis. So water moves from areas of high concentration to areas of low concentration. Please understand that when we're talking about the tonicity a cell is in, we define tonicity by the concentration of solute, not by the concentration of water. So when water is higher in concentration outside the cell, the solute concentration will be lower in concentration. And then water will move across the membrane and into the cell. Any question about any of that? All right, so cells can find themselves in 3 different environments, and we call these 3 kinds of osmotic solution. There's isotonic, hypotonic, and hypertonic. You should note that these terms are relative terms, so it is best or correct to say the solution outside the cell was hypertonic to the solution inside the cell. Any question about any of that? All right, isotonic is the easiest one to discuss, and that's just when the concentration of water is equal in concentration outside the cell as it is inside the cell. The cell is happy in that case because there's no net movement of water into or out of the cell. What do we mean by no net movement of water? Does that mean that no water molecule will ever move into the cell if the cell is in an isotonic solution? Come on, people, that's yes or no questions. You have a 50-50 chance of getting it right. Let me see.
Will they just be evenly distributed, kind of like equilibrium?
Yeah, it's an equilibrium. For every water molecule that moves into the cell, we'll have 1 water molecule move out. So that's what we mean by there's no net movement of water into or out of the cell. It doesn't mean specifically that no water molecule will ever move in. It's just that when we're talking about the total number of water molecules inside the cell, there will be no increase because the solution is isotonic. Come on, switch. All right. Hypotonic solutions is when the solute concentration outside the cell is lower than the concentration of solutes inside the cell. And the water is higher in concentration outside the cell than inside, so water will move into the cell. Note that the tonicity is defined by the solute concentration, but it's the water that's doing the moving. So if we're in a hypertonic solution, water will move into the cell, and that will cause the cell membrane to swell and expand. If the cell membrane doesn't have a cell wall to come up against, the cell membrane will expand until it pops, in which case the cytoplasm will leak out and the cell will die. If, on the other hand, the cell has a cell wall, the cell membrane will expand up until it touches the cell wall, and then the cell membrane cannot expand any further. That will limit the water coming into the cell. No more water will come into the cell. We're talking about net movement, no more net movement of water coming into the cell. And that's how the cell membrane can protect a cell. Why, if you've ever seen Elodea, a plant-like protist growing in water, why it can grow in water, because the cell has a cell wall, and when water moves into the cell membrane will expand up into the cell wall, and then the cell wall protects the cell, stopping the movement of water into the cell. And that's actually how penicillin kills cells. It damages the cell wall in the bacteria, and most bacteria, when they're infecting us, are in a hypotonic solution, and then water will move into the bacteria, and the cell membrane will expand up into the cell wall, but the penicillin is around and it damages the cell wall, so there's holes in the cell wall, and the cell membrane can protrude out of that hole. More water can move into the cell, and that'll protrude the cell membrane even bigger out of that hole until the cell membrane pops. And that is called osmotic lysis, which will kill the cell. All right, any questions about hypotonic solutions? The next is a hypertonic solution. That is when the solute concentration outside the cell is higher than the solute concentration inside the cell. If that's the case, then water will move from inside the cell to outside the cell. This will lead to shrinkage of the cell membrane, and the cytoplasm will shrink, causing plasmolysis as the cell membrane starts shrinking away from the cell wall, and it will just shrink and shrink and shrink until it shrivels up. Plasmolysis generally leads to cell death. It does depend on how hardy the cell is, but cells like E. coli, plasmolysis will kill them. And if you don't know, this is the reason why molasses can stay at room temperature for years. And I know it's years because, let's see if I get this right. I made some molasses candy for my father, and we figured out the bottle of molasses was at least 20 years old. And it had certainly been opened, and bacteria from the air fell in it, but that bacteria died because the water from inside the cell left the cell, causing the cell membrane to shrink and pull away from the cell wall, and that killed the cells. Why the molasses was good for 20 years, and my father did eat the molasses candy and enjoyed it greatly, and it did not kill him. He's dead now, but he died of, well, heart disease. But that was many years after I made the molasses candy. The molasses was 30 years old before my mother said, I should probably throw this bottle away. And it looked fine after 30 years, so it probably was fine. Just because of molasses being a hypertonic solution. Any questions about tonicity? If not, let me delete that because that's not where we're going to stop and go on. The passive processes are okay when the concentration of a substance required by the cell is higher outside the cell than inside the cell. Like if a plant needs water and it's in a pond, there will be more water outside the cell than inside the cell. But what about when a cell is in an environment with a low nutrient concentration, such as a cell in the ocean? If you don't know, the ocean is a desert, very little nutrients and actually very little life in the open ocean. And then a cell in the desert, like the desert sands of the safari. How does the cell survive in such places? Well, the cell can't rely on a passive process, so it uses an active process to move the material into the cell against its concentration gradient, and this requires transport protein and the use of ATP to give the energy the cell needs to move that molecule from outside the cell to inside the cell. All right, so moving a molecule against its concentration gradient requires the cell to spend energy. Usually that energy comes from the cell grabbing it from ATP. It also requires a transport protein, a protein that's in the membrane, and this protein is generally molecule specific, meaning we have a protein for fructose and we have a protein for glucose, and we have a protein for maltose, and we have another protein for all the other sugars you can think of. All right, and that's active transport because the molecule is moving against its concentration gradient. In prokaryotes, we have a special form of active transport. It is called group translocation. This is only found in prokaryotes. It is not found in eukaryotes. Group translocation requires a transport protein. It also requires a high energy phosphate compound like PEP, and that's similar to ATP. The high energy phosphate compound gives the cell the energy it needs to move that molecule against its concentration gradient. And then the molecule which is transported, such as glucose, is chemically altered by the group translocation. So let's say we're moving glucose into the cell, and there's already 5 molecules of glucose inside the cell, and there's 1 molecule of glucose outside the cell. The cell is going to want that last glucose molecule. And if it's a prokaryote, it can use group translocation. And prokaryotes can use active transport as well. But here we're talking about group translocation. And the glucose is moved into the cell using PEP and in a active transport protein. But the phosphate in PEP, when it comes off of PEP, that phosphate goes to glucose. So glucose is chemically altered in active transport. Let me shut off my phone here. Sorry about that. Trying to shut it off. There we go. It's thought that the alteration of the chemical moving into the cell, like glucose, makes it easier for glucose to move into the cell. For example, let's say we have 10 molecules of glucose already, no, that wouldn't be right. Let's say we have 10 molecules of glucose already inside the cell and 10 molecules of glucose outside the cell. The cell is going to want to pump all of those glucose molecules outside the cell to inside the cell, but it's going to be against the concentration gradient, so we need an active process to move it into the cell. Well, in prokaryotes, we have two ways to move the glucose into the cell. The first is active transport, and some of the glucose will move into the cell with active transport. So that'll cost the cell 1 ATP molecule for each glucose moved into the cell, and we're just going to roll it down the middle, which is actually what the cell does anyways. It will take 5 of those glucose molecules and move them into the cell by active transport, costing the cell 5 ATP. Okay. The other 5 molecules of glucose will move into the prokaryotic cell by group translocation. Group translocation does require a transport protein. It doesn't use ATP. It uses a molecule like PEP, which supplies the energy that the transport protein needs to transport the molecule of glucose against its concentration gradient. Okay, so that's all the same. And then the phosphate from PEP will be transferred onto glucose as the glucose is being translocated into the cell. So while it's being moved into the cell, the phosphate group will be added to glucose, changing glucose into what used to be glucose will change into glucose 1,6-phosphate. And so now inside the cell, we will have 15 molecules of glucose and 5 molecules of glucose 6-phosphate. It is easier for, let's say, the next molecule of glucose to be moved into the cell, because instead of having 20 molecules of glucose inside the cell, we have 15 molecules of glucose and then 5 molecules of glucose 6-phosphate. And that is why they think that prokaryotes have group translocation, just to make it easier to move a large number of molecules inside the cell when it wants to move all of them into the cell, like the glucose. Any question about any of that? All right, if not, let's move on. Let me take a look and make sure I'm doing well on the time. Yeah, we go to close to 6:20, 6:15. Somebody can tell me at 6:15, and then I'll stop if I don't notice myself. All right, where am I? There we go. Let's talk about the cytoplasm of prokaryotic cells. It is the substance inside the cell membrane. It is mostly water. About 80% of the cytoplasm is water. The other part of the cytoplasm is dissolved solutes in the water. And that can be dissolved protein, dissolved DNA, dissolved ATP, dissolved GTP, meaning nucleotides. It could be dissolved amino acids, dissolved glucose. Everything the cell has in the cytoplasm that is dissolved in the cytoplasm. The nucleoid region is a part of the cytoplasm, but meaning there's no distinction between the nucleoid region of a prokaryotic cell and the cytoplasm because there's no barrier. There's no membrane from the nucleus, nuclear membrane. There's no nuclear membrane. The nucleoid region is simply wherever the DNA is floating in the cytoplasm. We also have ribosomes and inclusions, which are inside the cytoplasm. But if you remember, I mentioned they are not dissolved in the cytoplasm. A ribosome is big enough that it does not go into solution. And you can actually see it here in this artist rendition, or if I can blow this up, the little dots over here, those are ribosomes. An inclusion is also not dissolved in the cytoplasm. The inclusion is something the cell has been accumulating, and there's so much of it, it precipitates out of solution. So the inclusion is, I guess you'd call it a solid structure, maybe crystalline, which has come out of solution, giving rise to the inclusion. All right, any question about the cytoplasm? I've already talked about the nucleoid. I should mention the nucleoid region is where the chromosome is located, not necessarily the plasmid. And the chromosome is dissolved in the cytoplasm. And wherever it's floating around, that's where the nucleoid region is. It does stain differently, why it's called the nucleoid region. In eukaryotes, the DNA is in the nucleus, so it's inside the nuclear membrane. And there is a distinction between the nucleus and the cytoplasm. But in prokaryotes, there is no distinction. The nuclear region is a part of the cytoplasm. It just stains differently, and we call it a different region of the cell, but it is part of the cytoplasm. Also in the cytoplasm, you can have small molecules of DNA, and we call those plasmids. You can find plasmids in many, but certainly not all, prokaryotic cells. And you can also find plasmids in yeast. We don't really have plasmids in human cells, although we do have extra chromosomal DNA. And I'll try and talk about that a little bit at the end of this lesson. So probably not today, but in our next lesson. All right, I've already discussed the ribosomes. They are the portion, where the facility where DNA is synthesized. They are a very large protein and RNA complex, meaning that ribosomes are made of both RNA and ribosomal proteins. And they're big enough they do not go into solution of the cytoplasm. You don't need to know that ribosomes have subunits, but you do need to know that prokaryotic ribosomes are 70S in size, and that is smaller than eukaryote ribosomes, which are 80S in size. All right, let's talk about inclusions. Inclusions are reserve deposits that the cell has been accumulating, and at low concentration, the reserve deposit will be in solution of the cytoplasm. However, the cell wants that material, and so it'll pump it into the cell, and there will be more and more of it in the cell. Eventually, excuse me, eventually the material will be so high in concentration that the material will precipitate out of solution, in which case it will form an inclusion. So an inclusion is just material the cell is accumulating, and there's so much of it, it comes out of solution, forming the inclusion. There are different inclusions, and that's all you need to know. You don't need to know the name of them. You just need to know there are different inclusions. It depends on what the cell is, what type of an inclusion it has, or what kind of inclusion it will have. Here are 3 inclusions just to show you some. Magnetosomes, something that's magnetic, sulfur granules, and these inclusions, this must be a sulfur metabolizing cell, and it just accumulates a whole bunch of sulfur inside it. And there's so much sulfur being accumulated, it precipitates out of solution. And these cells have a lot of sulfur in them. So they have a lot of material that they can use for their metabolism. And then metachromatic granules shown here. Let me see if I can blow that up a little. The cell is drawn in yellow. So these are rods, and it has 3 inclusions. I mean, yes. Yeah, the cell is shown in yellow, and this cell has 3 inclusions. Any question about any of that? All right. The next cell structure I want to talk about is endospores. Endospores are a specialized resting cell that is produced only by certain species of bacteria. The species that produce endospores is mainly the 2 families Bacillus and Clostridium. It is true there are a few other species which are not Bacillus and Clostridium, but most of the endospores are made by Bacillus and Clostridium. Endospores are a highly resistant cell. They're resistant to desiccation, resistant to heat, resistant to toxic chemicals, they're resistant to radiation. And so these cells can survive conditions that the vegetative parent cell cannot survive. I used to know the longest living endospore. It was from a drill in Antarctica, and they found it like 40 feet down in the ice. Anyways, they took it out and determined its age, and it was something like 30 or 40 million years. Is that right? Or was it 300 or 400? I think it was 30 to 40 million years. And they got that endospore to germinate, and then it went on and became a vegetative bacteria cell. And then that vegetative bacteria cell started dividing. The point is that endospores are highly resistant, and they can survive extreme environmental pressures or extreme environmental conditions. And that's why the cell makes the endospore. It will only make the endospore when the environment is deteriorating and the cell says, oops, the environment's getting bad. I might die. I better make an endospore. And here, these white areas of the cells here are endospores where they're actually inside the cell. These endospores are outside the cell. And what happened is they've not germinated, but they've come out of the parent cell which made them. Any question about any of that? All right, sporulation or sporogenesis is the formation of endospores. Like shortly before this Christmas, I grew up some bacteria cells from endospores, and germination is the term where the endospore germinates and forms a vegetative parent cell, which can then go and do metabolism and whatever the parent cell normally does, meaning its function. And then when the environment deteriorates, the parent vegetative cell will start making a spore inside it, and that's called endospore formation or sporulation. Any question about any of that? All right, here's an actual endospore. You can see it's very tough. And then this is the parent cell that's making it. Let's talk a little bit more about endospore formation. And the cell goes through a cell division. So the DNA or the chromosome replicates, and then one chromosome moves to one side of the cell, and the other chromosome moves to the other side of the cell. This one doesn't need to move much, but it does move to this side of the cell. And then the cell divides into two. But note that this division is not binary fission. The cell is not dividing evenly into two. One side will stay the vegetative cell, and it'll be a larger part of the cytoplasm. The other cell or the other side is going to go give rise to an endospore, and it's the smaller cell of the two daughter cells. All right. So the cell will go through division, and what happens is in cell division, the cell membrane forms between the endospore forming property, meaning the endospore forming area, and the vegetative area. The two cells are now divided, and they're essentially two daughter cells. It's not called that because this isn't a real cell. It's an endospore, which is a cell, but it's not metabolically active. Come on, mouse, where are you? It's not metabolically active, and it won't do anything until it germinates and gets out of being an endospore. The spore septum then forms, and that makes two membranes around the endospore. Peptidoglycan layer is then put in the two inner membrane regions. So in a sense, this cytochrome C, is that what the term is? Let me see. Yeah, I'll just skip it. So there's like a cell wall forming inside the two plasma membranes, and then the spore coat will be added. That'll be the outermost part, and there you can actually see the spore coat there on this endospore. And then the last thing the cell does is it lyses the cell, and that will release the endospore. See if that works. Well, it's not opening when I right click, so let's try left clicking. See it. Try that again. Oh, there it goes. It's not that critical of a video, but let's move on since I'm about to stop. And then we'll watch the video on endospore formation. Did I stop sharing my screen? Sorry, I didn't mean to do that. Here we go. And you're not seeing this, so let me share my screen. So this cell is going to form an endospore, and you see it forms mainly on one side of the cell. But if you look at that very closely, oops, right there is a little bit of the parent cell on this side. So that endospore is inside the parent cell. Any questions about endospore formation? Oops. All right, let's close that down. Oh, I should mention that endospores are not a way the cell can grow. They're not a reproductive structure. And you can actually see that one vegetative cell makes one endospore, and then the vegetative cell will die and the endospore will continue. One cell makes one endospore, and then one endospore will germinate to make one parent cell or vegetative cell. So it's one all along. There's no reproduction in here. Any questions about any of that? Let me see. I think I'm supposed to finish chapter 4 today, in which case we won't do it, but I'm going to probably skip. No, I don't have to finish chapter 4 today. I have it on the syllabus for Tuesday of next week. So I guess we will be discussing the structures of eukaryotic cells. Let me go ahead and talk about the first two slides, just to talk about eukaryotic cells and their cell structures and their membrane-bound organelles. And after that, we will stop talking about them, meaning I'll stop for the lab. Come on, that didn't work. I'll get this out of the way. So here we're talking about eukaryotic cells. Can't quite get there. There's a good place. And they have what the book calls organelles, or membrane-bound organelles and non-membrane-bound organelles. The organelles are in or membrane-bound organelles are in the red boxes here. Make sure everything is correct. And the others, like the microfilament and the microtubule, the cytoplasm, I don't call them non-membrane-bound organelles, but you could. I tend to call them cell structures. That a membrane-bound organelle is an organelle that has a membrane about it. Come on. Anyways, on this side of the cell, we have an animal cell, and it tends to be roundish. On this side of the cell, we have a plant cell, and it has this particular shape. This one looks like it's one hexagon for this one. 1, 2, 3. And that's the shape of the cell wall determines the shape of the plant cell. Most of the membrane-bound organelles are found in both animal cells and plant cells. I can think of one exception, and that is the chloroplasts are found only in some plant cells. They're not found in any animal cells because animals can't undergo photosynthesis, and photosynthesis occurs in green plants inside the chloroplast. You can notice how I qualified that. I'm not talking about the parasitic plants, which are not green, and you don't need to know about those, but there are up to a dozen of them. Anyways, animal cells have no chloroplast, and some green plant cells do have chloroplasts, and those are cells that engage in photosynthesis. What plant cell does not have a chloroplast? What plant cell does not have a chloroplast?
Is it algae?
No, algae is actually a protist, but it's a plant-like protist, and they do have a chloroplast. Good guess, but no. I'm talking about a green plant cell. What part of the plant will not engage in photosynthesis? I can't believe somebody doesn't know that one. Obviously, the leaf is green, so it's engaging in photosynthesis. What part of the plant is not green?
Is it the root?
Yeah, the roots. The roots don't ever see sunlight, so they don't have chloroplasts. And if you ever pull up the root, they're also not green. Now, a stem could be green, in which case it is photosynthetic, but the stem may not be green, in which case it is not photosynthetic, and like the bark of a tree is not photosynthetic. And we won't talk about stems, but roots, all roots are not photosynthetic, and they're not green, and they lack chloroplasts. So in all plants, some regions of the plants have chloroplasts. Those are the photosynthetic cells. And then the non-photosynthetic cells lack chloroplasts. That's the main organelle that you find in plants and do not find in animal cells. There is actually another one. It's not labeled appropriately here, and that is the central vacuole. Come on. Is found in plant cells, but it is not found in animal cells. The central vacuole is the largest membrane-bound organelle in a plant cell. We do have vacuoles in animal cells, but we do not have a central vacuole. A vacuole is a small organelle. Let me see if I can find a small one. This would be close to 1. And this would be close to 1 too. And that would be close to 1. These are not vacuoles, but they're about that size, okay? And that would be that same size in a green plant cell, because green plants have a central vacuole and they have vacuoles. There is one other cell structure I want to talk about, and that's the centrosome. You find it in animal cells. It is missing in plant cells. The centrosome has 2 parts, the centriole and the centriolar material. Let me see if I can blow that up. It's important in, I can't blow it up further. Let me see if I can end it and see if I can get it bigger that way. There we go. Centriole right there. It's important in animal cells in making the mitotic spindle, which is seen in mitosis and meiosis. Curiously, in green plants, they do have a mitotic spindle, but they do not have the centriole, which makes it. I guess I'm only going to talk about this slide here. Are there any questions? If not, I'm going to end it here and I'll see you at 6:30 for the lab. Oops, wrong one. All right, goodbye.